Hello everyone and a warm welcome. In this lecture, you will get to know about history of microbiology. You will study who is father of microbiology, what are Koch postulates, and what is contribution of Louis Pasteur and other scientists. This lecture is useful for beginners of the subject microbiology as well as for the other subjects like botany, zoology, for the students of other courses like biotechnology, medical lab sciences, for physiotherapy as well as for the nursing. In this lecture series, in this channel, you will find interesting and fascinating subjects. So, subscribe to the channel. First of all, what is microbiology? This is actually the branch of the main subject, which is biology. In microbiology, we study the organisms which are so small that they cannot be seen with the naked eye. If we want to visualize them, we want to see them, we require the help of the equipment called microscope. So these organisms are called microbes or microorganism. Their study is called microbiology. The microbes or microorganism, they are minute living things. They are minute living organism which we are unable to visualize with the unaided eye. That means we require the help of the equipment called microscope. These microbes can be classified as bacteria, protozoa, viruses, yeast and molds, and some parasitic worms also. This is showing the world of microbes. There are some bacteria shown here, some fungi, protozoa, viruses, and the algae. This is a very interesting branch of the biology in which we are going to study about these invisible creatures. Some of these are helpful, beneficial for us, while some they cause the diseases. So are all microbes too small? Yes, they are small, but there are some exceptions also. Some are quite large. They are so large that we can see them with the naked eye. You can recall that if you keep a piece of bread in the open, in the damp, and then after some days there will be some growth on it. You can see some black spores. These are of the fungus and we call it as the bread mold. Then there are some filamentous algae which we can see with the naked eye. And now there is a recent news in which some bacteria, they can be seen with the naked eye. They are so big that we don't require a microscope. We can see them with the naked eye. These are Thiomargarita and Pulopycium. This is showing the image of Pulopycium cell. This is showing Thiomargarita nambiensis. They are too big and they can be seen with the naked eye. First of all, we will study about the discovery of microorganisms. Lustaticus and Fracastrum, they can be considered as two physicians who proposed that disease can be because of some invisible living creatures. Before them, the concept was that if a person is suffering from a disease, then that is because of the wrath of God. That person has done some sin and the God is punishing him in the form of the disease. Antonie Van Leeuwenhoek can be considered as first person to observe the microorganisms, and he not only observed them but he described them very accurately. Antonie Van Leeuwenhoek, because of his discovery of microorganism, can be considered as father of microbiology. Basically, he was a Dutch merchant, but his hobby was to grind lenses, and by grinding the lenses, he prepared some combination which were the earliest microscopes, and through this combination of the lenses, which now we can call it as the microscope, he visualized certain materials. So what were those materials? Leeuwenhoek observed material like teeth scraping, rainwater and peppercorn infusions. When he observed these material under his combination of the lenses, the microscope, then he also drew those patterns. His drawings can be seen here, and he not only drew them but he also described them. And today we know that what he described, those were protozoa, algae and even some simple bacteria. Since they were small and the term microorganisms was not known there, so he called them as the little animals and he gave the term animalcules. Before Leeuwenhoek, there was a remarkable discovery which was of Robert Hooke. Robert Hooke also used some microscope and he observed these cells. Those cells were from the bottle cork. Actually, what he observed were just the non-living cells, and he gave them the word cella, cella meaning a small room. He not only found this cell, but he also communicating his findings in Micrographica, and he was the person who led the foundation of cell theory. These were the cells, the compartments which were observed by Robert Hooke in the bottle cork. There can be some other theories which are related to the origin of life, which are also the landmark discoveries, and they led to the development of the subject microbiology. So we will also study the origin of life and different theories related to it. The concept was of the Aristotle, who was a Greek philosopher. He gave the concept of the spontaneous generation, which is also called abiogenesis. According to him, the living organisms, they arise from the non-living matter. Like a rat arises from some matter like the dust, from the other matter like some other. The concept of spontaneous generation was opposed by the different scientists like Francesco Redi. He performed an experiment. He took 3 glass jars and kept a piece of meat inside them. The first jar was the open jar. Flies came inside the jar, they laid the eggs formed maggots developed into the flies. The second jar was covered with a piece of cork. Flies came, but they only laid the eggs here. In the third jar, it was covered with a cloth. The flies came, they laid the eggs there, and these eggs they developed into the maggots. So he gave the concept that the flies, they arise from the maggots but not from the meat. There was the Golden Age of Microbiology, in which there was the contribution of Louis Pasteur. Louis Pasteur gave important discoveries, like he was able to prove the relationship between the microbes and the disease. He also gave the concept of immunity, and later on the antimicrobial drugs were also discovered by the other scientists. So majority of the work was done by Louis Pasteur, then other eminent microbiologists who led to the development of this field microbiology. The period from 1857 to 1914 can be considered as the golden period of microbiology. So first of all, coming to the contribution of Louis Pasteur. Louis Pasteur has contributed a lot to the subject microbiology. So we also call him father of modern microbiology. Remember, father of microbiology is Antonie Van Leeuwenhoek, while the father of modern microbiology is Louis Pasteur. He gave the germ theory of disease. He also formed the germ theory of fermentation. He laid the foundation of aseptic methods. He also disapproved theory of spontaneous generation by performing the Swannick experiments. He gave the germ theory of disease. This theory was earlier proposed by physicians like Pasteur, Gallen and Basset. Then this theory was further extended by Robert Koch and Louis Pasteur. According to germ theory of disease, the microorganisms, they are known as pathogens or germs, and they can lead to disease in animals as well as plants. Like the disease which is a disease pebrine in the silkworm, that was known to be because of the microorganism. Then potato blight disease, it was discovered to be because of certain fungi. In the aseptic methods, the Louis Pasteur performed the experiments in which he took the S-shaped flask and he let the air inside them. He gave the concept of the aseptic techniques. In the first one, when the broth was added, this liquid was added, it became turbid. In the second one, this was the shape S-shaped and it was also heated from beneath and also from the intermittent heating at the neck of this swan shaped. So this liquid, it remains sterile, it didn't become turbid. So this led to the final disapproval of the spontaneous generation. In the germ theory of fermentation, Louis Pasteur described the souring of milk. He was the first person who told that the milk becomes sour because of certain microorganism. Then he also told that there are certain yeasts which are responsible for the formation of alcohol from the sugar. He used the different fruit juices to form the alcohol using the yeast. According to him, there are certain microorganisms which are present in the air as contamination. So they can cause the fermentation of the milk as well as of different sugary substances, and those substances, they turn to be sour because of the acid formation. Louis Pasteur also gave the technique which we widely use these days. This is called pasteurization. This is a technique, the method by which we heat any of the liquid at a temperature of 62.8 degrees Celsius for 30 minutes. This heat treatment destroys all the pathogenic microorganisms. This is still used for the industrial production and the packaging of certain milk and the milk products. Louis Pasteur also gave number of vaccines. Although the first vaccine was given by Edward Jenner, Edward Jenner was the first person to give the vaccine for the smallpox. But he couldn't explain the mechanism why there is protection, and the term vaccine was given by Louis Pasteur to honor Edward Jenner. Louis Pasteur formulated the vaccines for the disease like fowl cholera, for the anthrax, and also for the rabies. Now there is a little food for thought. Think for this question and do answer in the comment section. The question is, why did Louis Pasteur make an S-shaped neck for his flask? The option A, so that the air could escape. Option B, so that the microbes would be collected. Option C says to help the microbes reach the fluid. Option D, so it was easier to hold. Write the answer in the comment section, and if you want further clarification, you can tell me. So the answer is so that the microbes would be collected. The answer is the option B. Next contribution was of the Joseph Lister. For his contribution, he is also called father of antiseptic surgery. He was a physician, he was conducting certain operations, some surgeries, and he gave the concept that if the phenolic substances are used for the purpose of dressing, and if he himself and his students, they used the phenol to dip their hands, the gloves, in the solution, then the infections, they decreased during the surgeries. So he gave the concept of the use of phenol, and he also gave the concept of the sterilization of the instruments during the surgeries. And because of this, there was a remarkable decline in the infections which used to be following after the surgeries. Next contributor is Robert Koch. Robert Koch is also called father of medical microbiology. So this one is the fourth father. First one was the father of microbiology, Antonie Van Leeuwenhoek. Father of modern microbiology, Louis Pasteur. Father of antiseptic surgery was the Joseph Lister. Now the father of medical microbiology, Robert Koch. He discovered the causative agent of tuberculosis, Mycobacterium tuberculosis. He also discovered the causative agent of anthrax, Bacillus anthracis, and he gave the concept of the disease and the microorganism, which is known as Koch postulates. He also developed the media for cultivation of bacteria, which are the nutrient broth and nutrient agar. What are Koch postulates? The first postulate says that if an animal is diseased, then the suspected pathogen should be present and we should be able to isolate those pathogens. The second is that pathogen should be isolated in the pure culture form. The third postulate says that the isolated organism which has been grown in the pure culture, if it is introduced in the healthy animal should get the disease. The fourth postulate says that from this diseased animal, the organism should be re-isolated and it should be original, it should be the similar to the first one which was isolated from the first diseased animal. There are some other scientists who also led to the development of the subject microbiology. There was Fanny Hesse, who gave the contribution the use of agar as a solidifying agent. Bejanik, he was the first one to isolate the tobacco mosaic disease agent, the filtrable agent which was given the term virus by Louis Pasteur. Walter Reed, he discovered the agent for the yellow fever, and he also proved that yellow fever is because of the bite of the mosquito. Charles Chamberlain was the first person to discover the bacterial filters and also the autoclave. Dmitry Ivanovsky, he was able to isolate to obtain this filterable agent which is the virus. Todd and Hurley, they isolated the bacterial viruses, also called bacteriophage. Behring and Kitasato, they discovered diphtheria antitoxin. Alimatnikov discovered phagocytosis. Paul Ehrlich, he gave the drugs arsenic drug salvarsan and proved it to be effective in the treatment of syphilis, and the term magic bullet was given by him. Alexander Fleming, Florey and Chase. Alexander Fleming was responsible for the discovery of the antibiotic penicillin. Along with Florey and Chase, they were responsible for the application of the penicillin to the patient for the clinical trials of penicillin. Vinogradsky discovered the bacteria which could oxidize iron, sulfur and ammonia to obtain energy. He also discovered anaerobic nitrogen fixing bacteria. This is all about the history of microbiology. Hope you have liked this lecture. Like, subscribe and share among your friends. For further updates, keep watching. Thanks for your patient hearing and best wishes.